Hello from Inside Drug Discovery. In this video, we will explain why drug discovery does not end with the initiation of clinical trials. We will also define some of the common toxicology studies that are performed in parallel with early clinical trials. Once lead optimization is complete and a compound has been advanced into clinical trials, one might think that the drug discovery phase is complete. After all, the goal of drug discovery is an IND approval and kicking off drug development in the clinic. Drug discovery is not finished, however. While lead optimization is indeed complete, assuming the investigational drug performs well in the clinic, preclinical safety studies will continue to be performed. For most early clinical trials, volunteers and patients undergo relatively short-term dosing regimens with the investigational drug. The emphasis is on safety and early evidence of efficacy. And these studies are typically rather short. The preclinical safety studies that support early clinical trials are also relatively short and mostly emphasize acute toxicity of the investigational drug. If the drug is intended for chronic use, think of a blood pressure drug or hormone replacement therapy, then preclinical safety studies must study the long-term toxicity risks of the compound. We will focus on two long-term risks: carcinogenicity and developmental toxicity. These long-term studies are typically performed concurrently with early clinical trials. This concurrent activity is why some people favor the term non-clinical research rather than preclinical research for drug discovery, because the research window for drug discovery is not mutually exclusive of the window for drug development. They overlap. What are the animal toxicology studies that often overlap with clinical trials? First, we have oncology studies. These are typically 2-year studies performed in mice and rats. So basically, you are dosing an animal for nearly its entire life with a drug to observe carcinogenicity. Before these studies are completed, the longest preclinical studies will likely involve 6 months of dosing. So the longest allowed dosing regimen for patients in the clinic will also be 6 months. Longer studies will not be allowed without completion of the carcinogenicity studies. Similarly, developmental and reproductive toxicity, sometimes called DART, studies must also be completed. These studies focus on toxicity towards a fetus and potential reproductive risks for patients. They are generally performed in rats and rabbits. A particular point of interest with these studies is their direct connection to gender differences among patients. Without the completion of DART studies, women of childbearing age may be excluded from clinical studies for the safety of a potential fetus. This is a reasonable safety concern. Of course, exclusion of women of a certain demographic may skew efficacy and safety information on the drug. This is also a reasonable concern. Therefore, the later timing of DART studies is often a point of controversy. We have now covered the idea that some aspects of drug discovery operate concurrently with drug development. We have also described carcinogenicity and DART studies. I appreciate your watching the video. If you'd like to receive updates of new videos, then please subscribe to the channel. Take care.